Well, hello, everyone, and welcome to today's Research America Alliance discussion. Thank you so much for joining on Zoom and for your partnership in our alliance. If your organization is not a member, we do invite you to stay as our guest, but please get in touch with my colleague, Joel. He'll be happy to connect you and discuss the value that comes with being an alliance member organization. We'll be posting his email in the chat. Our special guests today are Daniel Pham, Cara Altimus, and Emily Baxi from Breakthrough Discoveries for Thriving with Bipolar Disorder, or BD squared. And it's fun to get to welcome Daniel back to Research America. He was a science policy fellow several years ago, and it's wonderful to see how his work and his interests are coming full circle. So the BD squared team will be speaking with us today about the important work that they're doing. BD squared is a collective force transforming what we know about and how we treat bipolar disorder. As always, please type your questions into the Q&A box or the chat, and we'll pose as many as we can during the Q&A portion at the end of today's session. So with that, thank you so much for joining us, and Cara, it's over to you.
Thanks so much, Tim, and thank you everyone for joining us today to learn about the new kid on the block. We are brand new. I believe today marks our 16th day, and in those 16 days, we have already posted several new funding announcements that we're excited to share, and really excited to share how they fit within a broader context and model of change, not only for bipolar disorder, but biomedical research as a whole. So I'm Cara Altimus, Managing Director of BD Squared, and I'm joined by Daniel Pham and Emily Baxi, my colleagues who are program directors. I'm going to kick us off and then pass it to them for some of the specifics. So to start, I want to introduce with some stage setting about bipolar. First, to share, 1 in 30 individuals are living with bipolar disorder. We also know that from the time of initial symptom onset to a diagnosis takes 7 years or more on average, and 50% of those individuals that receive a diagnosis will not respond well to currently available treatments. Overall, this leads to 10 to 20 years lower life expectancy for individuals living with bipolar. In our initial landscaping and work with the field, we very quickly understood that there were differences in funding and how bipolar was resourced. There are also very well described access issues across mental health. But at the root of many of those, we found that there were some real fundamental challenges in studying bipolar. One is bipolar is a heterogeneous condition with a very complex trajectory. It has several subtypes, but even within those, scientists and clinicians understand that there's a broad range of how bipolar develops and is presented. Additionally, research studies to date have struggled with scale, which is quite related to the heterogeneity. So as we started to think about how are we going to tackle and understand and bring real meaningful change to bipolar, we landed in a place of wanting to balance building out of the fundamental understanding of bipolar with a real acknowledgement that we needed to address the scale, heterogeneity, and trajectory of bipolar. I want to pause and say, while we're talking about bipolar, what I just described could be applied to many other disorders and illnesses, particularly those of the brain, but actually spanning a much larger area. And so as we've thought about the structure of these investments and how we build BD squared in these initiatives, we've built it for bipolar, but we also see this as a model of how we might reimagine and accelerate discovery and the movement towards improved care and treatments across biomedical research. So I want to pause and say we think, especially in the Research America space, we think a lot about how different kinds of funding are able to do different things. BD squared is philanthropically funded and incubated within the Milken Institute, Center for Strategic Philanthropy, where we really focus on bringing philanthropic dollars to to really complex problems, and in particular, science and biomedical research. Previous studies of philanthropy have found that it's only about 5% of biomedical research total. And so you might say, how much does 5% change an ecosystem? But when philanthropy is able to act and really harness the unique ways that it's able to fund, it's able to do something really different. So I wanted to pause and say, what is different about philanthropy, and how might we use that to design scientific initiatives that really harness and do something different, so that what we do might be complementary to work that the NIH or other organizations are doing rather than duplicative. So the first thing to highlight, not actually on this slide, is risk tolerance. Philanthropy is not moved by political votes. It is also not swayed by a need for stakeholder return on investment that allows philanthropy to play a long game that you might not see in other types of funding sources. As I mentioned, this is a little bit different. We have a philanthropic collaborative. And so we are not only thinking about philanthropy, but what does a collaborative of philanthropists do differently? And so a couple things there come to mind. One is scale. We're able to move faster and farther than you might if you were to go it alone. We have collaboration baked in at every step. So we have collaboration of funders, and we're asking for collaboration of researchers and clinicians, which we'll describe in a moment. We are bringing those various perspectives across our initiatives, and urgency is huge. We have people with lived experience at every level of decision-making, the funders through our scientific steering committees. And this real-world experience and a focus on bipolar is driving action very quickly within the organization. So with that, I'll introduce our 4 initiatives very briefly, and our theory of change, and then pass it to Emily and Daniel. So starting in our, you maybe can see my mouse, starting on the yellow box in the corner, the discovery grants are team-based grants focused on mechanistic research. We have animation. Moving clockwise, we have 2 platform programs that are, one focused on genetics and one on brain omics, both focused on building out fundamental knowledge of risk factors and therapeutic targets based on existing samples. And the last, as we move clockwise, the integrated network, which is going to be a platform of biological discovery within a clinical network. So to just touch on the theory of change very quickly, we have the pipeline, very simplified. So know that we're skipping some steps here of how traditional research results in changes in care. So we move from research, publication, clinical guidelines to changes in care. We've seen in the last decade a movement towards a model of a learning health network, which takes care and through sharing amongst clinicians and patients, the development of improved implementation and improved outcomes, which is able to lead to an update of the clinical guidelines and ultimately clinical care. We think about how we might put these together. We've added this deep phenotyping as a way to immediately start to understand the patients that are being cared for. And then BD squared starts to bring these 2 kind of opposite ends of the spectrum together. So discovery grants are operating in the mechanistic research space. We then put the genetics and the omics as a way to further connect what we understand about the basic biology and architecture of disease, and build that as a bridge to what we understand about individual patients that exist within the network. And the integrated network then sits anchoring real patients that we are engaging in research and care, so that we're able to combine deep phenotyping all the way through improved care. By having all of this under one umbrella, we see this as a real model of how we are going to accelerate basic discovery all the way into improved care for people living with bipolar today. So with that very high level overview, I'm going to pass it to Emily, who's going to dive in and share a little bit more about the integrated network.
Thanks so much, Cara. So as Cara mentioned, the integrated network is really designed to combine deep phenotyping and care improvement so that we're accelerating that journey and translation of research to the clinic. And the design was in direct response to the needs of the field, where we really honed in on this pressing need to understand the heterogeneity and the trajectory of disease, as well as the potential to predict changes in mood state and response to treatment after that. So to achieve these goals, we have committed an initial $75 million to building the integrated network. That includes the sites that will comprise the integrated network, as well as robust centralized services and core facilities to help bolster and really administer this. So that will include a data coordinating center, as well as a clinical coordinating center for all of the sites. The grant is anticipated to run over 5 years, and we're going to initially focus on bipolar 1, looking at longitudinally the changes in clinical, neural, and physiological markers. So finally, we really want to stress that the focus is really on care integration here, so that integrated network participants will be active participants in the network, receiving their own data and benefiting in real time from the knowledge and the insights that are generated through the integrated network. So with that, I'm going to hand you over to Daniel to take you through the next portion.
Thanks, Emily. Yeah, so as Cara mentioned, with the foundational programs, which I actually haven't seen an update yet, but so we know from the field and generally about bipolars, highly heritable. So, however, not much is known about the genetics of bipolar. So we put one of the programs dedicated to better understanding the genetics of the disorder. So this program allows for, I believe, a $10 million initial investment over 2 years for additional sequencing of samples from people with bipolar disorder throughout. The samples have already been collected, and we know that there's available, it's available in the field. So hopefully this will really accelerate our ability to better hone in on what are the genetic underpinnings that underlie bipolar, so that we can better understand the mechanism and the biology. Alongside that, we also know that to really better understand a disease or disorder, we really need to study the organ or where that disease may manifest. So the Omics, the Brain Omics program really focuses on getting better understanding of the molecular mechanisms, circuitry, and all that, using postmortem brain tissue of people with bipolar. So this is a program that is a $5 million investment over 3 years to focus on understanding the different types of omics. So that can include epigenetics, proteomics, even again molecular structures and circuitry in existing brain tissue. So that really, a lot of this data feeds into the deep phenotyping loop, like that you saw that Cara presented, to bolster the ability for the field to use this data to get at a deeper understanding of mechanisms that could lead to new interventions. And then that also will feed into clinical care. So the last program is the discovery grants. And these are really exciting in that it allows the field to deeply interrogate potential hypotheses that have been in the field for several years and get at a better understanding of how, again, these potential hypotheses could be causal or not. These discovery grants are interdisciplinary, and they are about 1.5 million over 3 years, so 1.5 million per year over 3 years for teams of 3 to 5 interdisciplinary researchers. And we've really built in the ability to also support the field, that one of the researchers also must be an early career researcher. And that collaboration is really a real priority in this program. So we aim to fund about 5 of these team grants, and then we'll also have 2 more cycles after this year to award additional teams that could, in the future years, be able to use the data that are coming out of the genetics, omics, and the integrated network to further interrogate additional, potentially new hypotheses in the field. So we are looking to build a diverse portfolio of different methodologies that could include neurocircuitry, brain metabolism, immunology, aging, and neuroplasticity. And I think these last 2 slides highlight the 2 governing philosophies that are, again, baked into the entirety of the project, the organization. Team science is something that we've been thinking a lot about. Not only are all of the 4 programs interconnected and have very interconnected leadership committees, but also there will be the ability to share and analyze data throughout the different programs in different ways, depending on if you're the researcher or clinician, that can really, again, accelerate findings and potentially interventions. And then I think the last slide is on open science, that again, really encouraging the data to be out there earlier and identifying best clinical practices and allowing more eyes on the data, so that again more will come out of this massive pile of data that we're hoping to gather in a really effective and responsible way. So I'll pass it back to Cara for any closing thoughts, and then I think we'll open it to questions.
Thank you, Daniel and Emily, and everyone for joining us to hear about BD Squared. We're really excited to be at the beginning of this journey and really committed to taking lessons learned from the community of many other biomedically focused organizations and scientific organizations to carry forth and create new discoveries for bipolar. Tim, should I stop sharing?
Either way, if there's a slide you'd like to leave up, or it's up to you. There may be things we want to reference back to, but let me just say thank you for the overview. That was fascinating to learn about what y'all are doing, as also some of the opportunities that you see on the horizon. I've got a couple questions myself, but I'd love to open it up to anyone in the audience who's watching. And we did have one question come in already. So let me ask that to the group. So will the clinical component also look at environmental factors impacting someone with bipolar disorder, like sleep, food, stress, those kind of issues?
I'll pass it to Emily.
Yes, so really our goal here is to have a holistic view of what is happening in the patient's life. So there will be comprehensive evaluations on a yearly basis, but in between, we'll also be tracking a patient's activity through lots of surveys and potentially wearables. So really trying to build as comprehensive an overview of what it is that they have experienced, lived through over this time period, and how that is influencing the course of their disease.
No, that's great. Anyone else want to add, or is that, I just don't want to cut someone off if anyone else wanted to jump in.
No,
okay.
I think Emily covered it, but that we are, while there's deep phenotyping and real clinical assessment, there's also an acknowledgement that with differences in trajectory and the heterogeneity, that we need real tracking in more real time, as well as the integration of clinical data. And so this integrated network really brings those pieces together in a unique way.
And Cara, while you're on that, some of this is just coming from my own my own inexperience, because a little of this went over my head and I don't know what the range of our audience is. I could try to define deep phenotyping. I'd make a guess at it, but if somebody would like to jump in and talk about deep phenotyping and exactly what that covers, as well as just what all is encompassed in brain omics, I'd love to hear what you're thinking about when you use those terms. It would help me in reflecting back on the conversation.
I love those questions. So starting with deep phenotyping, if we just kind of straw man, start with what happens when you go into a clinic. You are very quickly evaluated. You usually height and weight is taken, and let's say it's psychiatrically focused. There's an assessment of how well you feel and general perception, and then there's clinical notes, and that's roughly the start and end of what data we have on individuals living with a psychiatric illness. What we're talking about here, this deep phenotyping, is asking about, it's looking to get a much bigger picture of what's happening biologically. So not only will we collect the normal data that's being collected upon a clinical visit, we will also be taking biological measures, so blood samples, urine samples, and asking, what can we understand about the immune system? What can we understand about metabolism? We'll do brain imaging. So we'll ask, how is the brain responding across the trajectory of illness? And then, as Emily mentioned, we'll be collecting real-time data, so actigraphy, to understand how is a person's behavior changing. And so all of those data combine to create a much more robust picture of the human and the people that we're engaging with, so that we're able to really understand and provide more concreteness around what is intuitively understood as differences in the disorder. And then I'll let Daniel, who thinks lots about, so I don't talk the whole time, the omics, to talk through what that means and why it's so exciting.
Yeah, omics, I guess, is a terrible general word that combines the different types of ways you can look at the biology of a system, right? So when we say like genomics, means just the overall oversight of all the things in your genes. Proteomics is a word that describes, the like global snapshot of the proteins that are involved in a system. There's epigenetic, people say epigenomics, maybe, just like ways to how the global snapshot again of how your genes might be modified on top of just the blueprint itself. So we're really interested in all the just different molecular mechanisms that underlie what might give rise to bipolar disorder in the brain in a very in a biological sense. So it's been, I think I saw a question of, what are the biggest questions in the field. One of them is, what is the biological causes of bipolar? And this is one way that we can do this in a standardized fashion using a resource that's actually very underutilized in the field, which is human brain tissue. There's a lot of brain banks that are available for the research community that's really underused, and we're hoping through this platform to not only award somebody the ability to do this, but have a better understanding of what is out there. And we were able to start maybe even building networks and understanding what are the increasingly underutilized resources that we can make available to researchers to ask these really tough questions about the biology.
Well, Daniel, Cara, thank you both. That helps me a lot. I figure anytime I'm Googling while somebody is talking to find answers, that's a question we should make sure.
It's a flag that we should probably better clarify in our presentation. So.
No, I appreciate it. I just don't know what the split is between a clinical and a more and a broader audience. And Cara, you asked earlier if you should stop screen sharing. I think at this point, I see more questions coming in, and we won't actually need those slides. And this way, people can see you a little better. So a few other questions that have already come through. One here, sorry, some of them just disappeared. One, you talked a little bit about how you're integrating early career researchers into all of your discovery grants. Can you talk about how that involvement with early career researchers you found changes the value or the outcomes, or what's the push to make sure early career researchers are part of every grant?
Yeah, I can talk about that. One of the biggest barriers that we've seen in the field of bipolar is actually a loss of researchers in the field. So, as Cara had mentioned, in our funding landscaping, we've actually seen a steady decrease of funding from the NIH going specifically to bipolar, not in the context of other mental health disorders such as major depressive disorder or schizophrenia. So there was a huge gap of really trying to grow the field and just getting more early career researchers in there. So in the discovery grants, within the team of 3 to 5, the request for applications dictates that there must be at least one of the one of the lead PIs must be an early career researcher. And for that, we identify as being within 10 years of receiving their graduate degree. So that also allows us to keep the pressure on shortening the training time. So as we've also seen, postdoc times have increased, graduate school times have increased. So keeping that 10-year maximum can really help the field better change policy or support the early career researchers to move on. And also the last thing we've seen is young people bring really interesting, good ideas and improve innovation. So that is a huge piece of it as well.
No, that's awesome. Another question that came in is, will there be any attempt to cross-link large population studies such as NIH's All of Us or healthcare organizations or other groups into this work?
It's a great question. We've spent really time over the last year working to understand what measures are being collected across a number of large population studies, mostly focused on other brain-based illnesses, because that's where we think we will see the most alignment of measures, and then make real intentional connections in what we're measuring with some of the other large studies. Additionally, we're still in the process of identifying partners that will support the housing of data, but we are prioritizing those who will be able to efficiently connect the data collected in the integrated network to data collected from other studies and disease areas. So it's a actual scored component of our selection process for the data partner that will be involved.
Okay, there was a clarifying question then on just how your BD squared is functioning. You're obviously making grant awards,
Yes.
but are you is BD squared conducting its own research as well?
So I saw the question and I was like,
Yeah.
it's an important distinction.
Elaborate.
All research will be done with partner organizations. So we will have clinical sites that will be recruiting and conducting research and doing the clinical study. Importantly, the programs that Daniel talked about, the discovery grants, the brain omics, and the genetics, are grants for proposed research. The integrated network has been planned in collaboration with the scientific community over the last year. And so it is a developed protocol and will function more as a network so that we have the continuity of data across all of the sites. So while Emily, Daniel, and I are not performing that research, we have facilitated the development of the protocol and the organization so that we have standardized data collection across the work.
Okay. Another few questions that are related to some of those data challenges. And there's a kind of a series of these that I'll walk through. One, to what extent will you be examining family patient traumas that may have triggered bipolar disorder? And then there's some follow-up to go on to that.
I saw that. Emily, do you want to start?
Yeah, I can take that one. Thanks. So these are great questions, and really bring to the surface what we discovered in our own sort of surveying of the field and the needs. So this family trauma and history will be captured in the intake and then updated every year through interviews. But, I think the person who asked this question is really homing in on some things that are very much on our mind too, which is how do you make sure that you are capturing the diversity of this disease, and that those individuals who are most affected at this point of time may be the least likely to engage in something like an app and daily activity. So I think that's where, we don't have a perfect answer, but we want to make sure that we remain very much engaged with the patient community and those with lived experience, so that we are learning, what is it that would be the easiest way in which we can begin to track on a very regular basis how shifts in mood coincide with other changes in your life, and how do we benefit you and encourage you by maybe sharing parts of that data that may be useful and insightful. But I think, we all recognize this need, not just to survey a small portion of the bipolar community, but really a much larger segment to inform how care moves forward. And so, that will be done in conjunction with those with lived experience.
Okay, that's helpful. If people are interested in the grants and the work that are coming out, how can they learn more about potential enrollment or about how to get involved?
We have a website. It was, we'll put it in the chat, but bipolardiscoveries.org. And we have all of the RFAs and links to our grant portal are available there, as well as just more information about how we got started and our program board, the funders themselves, and their involvement with BD Squared. So lots on the website, and then Daniel, Emily, and I are always available.
Okay, great. Another question that came in. Are you planning on looking at promising genetic issues, such as genetic linkages between disease and epilepsy, bipolar, et cetera?
I'll start, and I'm sure Daniel will fill in more. We see this as extremely important. This is one of the reasons that the genetics program is one of the first ones to be up and out. Similar to the question about All of Us and other platform studies across populations, we also have prioritized the ability of the data that results from this funding to be linked and understood in the context of other illnesses as well. There is already really good data that shows linkages both with bipolar and other psychiatric illnesses, but more broadly an understanding of how brain-based illnesses or even health illnesses are related to one another in a genetic sense.
Yeah, and I'll just add that the way we wrote the Discovery Grant RFAs is really an encouragement of interdisciplinary research, right? So we do want people from historically have had experience in bipolar disorder to think about it as well in the context perhaps of what they've studied, and that again could really bring new innovations and decrease the silos that we all know we live in, especially in research.
No, that's great. And obviously there's a lot of interest in this because we keep getting good questions. So thank you all for being patient with all of this. How, if at all, are diversity, equity, inclusion, representation being prioritized in the studies that are being funded in the initial $150 million effort?
I love this question. Increasingly, we know that this has to be front and center in what we're thinking about across all of our RFAs. You'll see that this is a place that we are asking for commentary, and we've been very thoughtful about how do we even ask the question so that it is appropriate based on the scientific participation, the participants that are part of the research studies, so that we're thinking about this up front, and we're asking our partners in research how they're going to be a part of that and bake it into their work.
Yeah, and I'll also add, in especially the genetics and omics platforms, where we explicitly ask for information about demographics of the samples that they already have. We want to make sure any new data that come out is non-European centric, as we know that's mostly what's available now, and even just on a biological basis, that having more diverse samples will result in just better data. And especially if we're thinking about the genetics piece, thinking about rare diseases or like rare mutations that can occur in smaller families, that can come out of a really diverse sampling. So we do, and that's one of the ways we are going to evaluate the different applications is through that lens.
No, that's helpful. Another question that's come in. One of the criticisms of current bipolar care is over and under medication versus no medication, as well as reliability in patients taking their meds. Will you be tracking medication dosages, evaluating efficacy in the context of and controlling for the big picture of their environment?
Absolutely. Part of the power and really the impetus to build the integrated network as both research and care network was because we and the clinical community saw the necessity to bring these streams of information and knowledge together, that a person not complying with a prescribed medication is data in and of itself, and that we should be capturing that data to understand how does that, who is that, and what is the biology of the person that is not taking medication or is, and how does that affect their trajectory. And so really being intentional upfront about a dual priority of capturing real clinical information as well as the biological information, so that those two streams come together in an integrated way, hence the integrated network. Uh-oh, you're muted, Tim.
Oh, sorry. What kind of digital health tools are y'all looking at that may enable that collection and understanding of the data you're gathering?
I can take a first pass at that. So this is, one of the reasons we are able to conduct a study and a program like this right now is because of the availability of so much technological infrastructure that enables us. The first is really, as I mentioned during the slides, is this data coordinating center that's really going to be the crux of aggregating data from EHRs across different systems and bringing it together in one place so that it can be analyzed together and understood. But we also want to make use of surveys, sending them out rapidly to patients, so we are filling the gaps between visits. Understand, back to the earlier question about how do you ensure compliance and make sure that you are really engaging participants. We want to learn as we develop and deploy these apps, how is it that we can best get this information? What is the least burdensome, most rewarding way that we can do that? So technology, data, big data is going to be key to all of these movements, because truly what we're trying to do here is paint a picture of each of our participants so we can learn from their particular disease course, their particular biology, and then begin to understand, are there patterns in that help us inform better care in the future? So these are all really, really critical components.
Okay, that's great. And I will promise I am keeping an eye on the clock. We will wrap up on time despite all the questions. Can you speak a little bit? You're on, I think you said day 12, 16, very new, very new. BD squared is very new. What does the kind of trajectory look like for the next, we'll say 3 to 6 months in terms of what you expect to be able to accomplish? And
So while I joke, and it's not untrue, we launched 16 days ago. Daniel will tell you this is all he's thought about for more than a year, and maybe more than 2. So there's been a lot of planning that's gone into this launch moment that has really brought together the scientific community, has brought together the clinical community, has engaged patients throughout the process. So that has positioned us to be very ready to like jump off and get going. And so we've already posted RFAs across the 4 key programs. We have one more RFA that will be on the way. It's the site RFA for the integrated network. We're going to spend the next 6 months working with the community to get applications in, to evaluate those, to make our first funding announcements, and then be really focused on having successful teams and partners across the 4 programs. And then we hope, we plan next year to start the process again, but building on a foundation of teams and sites that are going and already learning, so that we're building on the 2 years we've been planning, and then almost a year of being around and alive.
And I'll just share.
Oh, go ahead, Daniel.
The RFAs for anybody who's in the audience are live now, and the deadlines are in October and November, or maybe November and December. That's right. So there's plenty of time. So these are the 5 RFAs that are we're really excited to be addressing and carrying out.
No, that's great. So looking maybe a little further out, can you speak a little bit about how the model you developed might be applicable to other fields on beyond bipolar? I know that might not be in the 6-month plan, but a little further out.
We plan to be a model to really show how, by bringing basic science and clinical science together in the same network, in the same community, we're able to accelerate the movement of bench to bedside. I think many of us in the biomedical community feel bogged down and hear often that it's an average 17-year time span from discovery to change in the clinic, or availability of a new intervention in the clinic. In brain-based science, you often hear that timeline is even longer. So what we're really targeting is the model that we're pulling these key pieces, we're building the data systems, we're building the cultural systems that allow the acceleration of findings that happen at the bench to move to bedside seamlessly. And so the real hope is that we're able to do this in bipolar, but that it's so successful that it becomes the model, that we start to see the piecing together of foundational science, hypothesis-driven science, and then platform study with patients built into a clinical network that allows that learning to be put in place and run.
That's awesome. Well, I hope you are successful. We will certainly be keeping a close eye and excited to just follow along. So on that, if people want to do one thing to follow up, to share ideas, ask questions, get involved, what is the one thing they should do to make sure they're in the loop?
Go to the website.
Go to the website, follow there. Okay.
You can sign up for our newsletter. Our emails are also available. The 3 of us are happy to chat or anything. So I know there are some extra questions that we didn't answer. So send them our way, and we're happy to address them.
Well, thank you so much. I know your website is posted in the chat for anyone who needs it, or just look for BD Squared. So with that, thank you so much to Daniel, to Cara, to Emily for all the time you've shared today. This has been a terrific discussion. We appreciate your thoughts, and we're looking forward to seeing what is next. So to everyone watching, keep an eye on your inboxes for information about our next Alliance discussion here at Research America. And we look forward to seeing you again soon. Have a great rest of your day. Bye for now.
Bye, everyone.
Thank you.